And do let me know if you are not able to hear it properly, right?
Why children do not look
हाँ जी, आवाज बिल्कुल क्लियर है?
Yes,
Yes,
miss.
ma'am.
Okay, okay.
exactly like their parents, or why some siblings look so different from each other? The answer lies in the process of meiosis, which generates the gametes for reproduction. Meiosis is the process by which haploid cells are produced from a diploid cell. For this to occur, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cell. Meiosis occurs in special cells called germ cells within the gonads of males and females. For the successful reduction in the number of chromosomes to occur in the new haploid daughter cells, 2 rounds of divisions are necessary. These divisions are termed meiosis I and meiosis II. Although the processes of mitosis and meiosis look similar, there are a few major differences. One obvious difference is that mitosis results in 2 diploid daughter cells, and meiosis results in 4 haploid cells. As in mitosis, meiosis begins after a cell has successfully completed the G1, S, and G2 stages of interphase. During the S phase of interphase, the DNA is replicated, producing 2 copies of each chromosome called sister chromatids. The paired centrioles in the cytoplasm duplicate and begin extending microtubules that will form the meiotic spindle. Sister chromatids remain attached at the centromere and condense as the cell enters prophase I of meiosis. Up to this point, the cell looks similar to mitosis, but 2 events occur in meiosis that do not occur in mitosis and lead to genetic diversity. The first event occurs during prophase I, when homologous pairs of sister chromatids lie side by side in a process called synapsis, forming a tetrad or bivalent. The homologous chromosomes share similar, but not necessarily identical genes. Once this structure is formed, the second event, called crossing over, can occur. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occurs, increasing genetic diversity. Prophase I concludes with the fragmentation of the nuclear envelope as the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend spindle fibers, forming the meiotic spindle. In prometaphase I, with the paired centrioles in place, the meiotic spindle is fully formed. The sister chromatids attach to the spindle fibers by their kinetochores. Here, another key difference between mitosis and meiosis occurs. Because of synapsis and crossing over in meiosis, homologous chromosomes remain aligned, so that a pair of sister chromatids is attached to only one pole by the kinetochore microtubules. In mitosis, each sister chromatid is attached to a spindle fiber. The fibers originate from opposite poles. During metaphase I, bivalents randomly align along the metaphase plate. Due to independent assortment, this alignment is random and adds to genetic diversity. In anaphase I, the homologous chromosomes separate and move toward opposite poles. Meiosis I ends with telophase I, when the chromosomes decondense and the nuclear envelope reforms. Cytokinesis separates the cytoplasmic material, and the 2 daughter cells are separated by a cleavage furrow. Since the final product of meiosis I is 2 haploid cells, meiosis II begins without the chromosomes going through another round of DNA replication. Centrioles again duplicate and begin moving to opposite poles of each cell. In prophase II, the sister chromatids condense while the spindle starts to form as the nuclear envelope disappears. In prometaphase II, the sister chromatids attach to the spindle by kinetochore microtubules, with sister chromatids attached to opposite poles. The spindle aligns the sister chromatids along the metaphase plate during metaphase II. During anaphase II, sister chromatids separate and individual chromosomes move toward the poles. The entire process ends with telophase 2, as the chromosomes decondense and the nuclear envelope reforms. Cytokinesis occurs, and cleavage furrows separate the 2 daughter cells into 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half its chromosomes from its mother and half from its father. Because men and women produce millions of gametes, and the selection of gametes in fertilization is random, this contributes to genetic diversity. This explains why a child isn't identical to either parent. But why aren't siblings identical? The diversity comes from several sources. In prophase 1, non-sister chromatids can exchange DNA through crossing over, increasing the genetic diversity of individual chromatids. In addition to crossing over, in metaphase 1, the pairs of homologous chromosomes align independently along the metaphase plate and sort independently into the daughter cells. This process, called independent assortment, produces 4 genetically distinct haploid gametes. As the total number of chromosomes increases in an organism, the number of genetically distinct gametes increases by 2 to the nth power. This means an organism with n equals 3 can produce 8 unique gametes. For humans where n equals 23, there are 2 to the 23rd power unique gametes formed, or over 1 million different possible combinations. The combination of independent assortment, crossing over, and the random pairing of gametes during sexual reproduction increases genetic diversity and explains why a child will not look exactly like his or her parents or siblings.
Okay, so was this video understandable?
Yes,
Yes,
please.
sir.
Okay. This was about, in fact, it was good that we all saw this video so that the entire process of meiosis 1 and 2 has been summed up in a comprehensive manner. And the last part of the video in which they were discussing about what are the factors that lead to genetic variation as far as meiosis is concerned. That's very important for you to remember. In fact, पिछली दफा भी मैंने last class में ये करवाया था, before the midterms, that why meiosis leads to genetic variation is because of random fertilization of gametes, because of millions of unique gametes formed, because of pairing of homologous chromosomes, crossing over, and independent assortment of the chromosomes at metaphase 1 and metaphase 2. And it is actually the independent assortment that leads to the different kinds of unique gametes or possibilities of unique gametes that can be formed. And which is why siblings don't look alike, and neither do the children are an exact copy of either of the parents, right? Acha. Now, having said this, we have to start this topic, which is gametogenesis. As the name indicates, gameto means gamete and genesis means to make. So we will be learning how sperms are made and how ova are made. For the sperm, the word spermatogenesis is used, and for the ova or the ovum, the word oogenesis is used. So let's first watch a video on spermatogenesis. You will watch this very carefully because I'm going to ask questions and we will have a discussion on it. So let's start.
Motion is
Oh,
what
sorry.
propels the sperm or causes the sperm to move. In this,
Acha. All of you can hear it properly?
yes, sir.
अच्छा ठीक है मैं थोड़ा सा आवाज बढ़ा देती हूँ. Okay, now watch it carefully. And also while you're watching it, relate your, in fact, connect the information that you have obtained while doing meiosis 1 and 2 onto this process. कि अब आपको imagine करना है कि ये सारा process जो meiosis 1 और 2 का है वो, how is it taking place within the gonads, which is the testis?
Video lecture, we are going to be studying sperm and their development. Okay, so just starting out with the sperm, and remember that sperm are the male gamete. And also be sure that you understand that sperm are also called spermatozoa. That's the same thing. So if you hear the term spermatozoa, we are referring to sperm. Okay, so let's look at the morphology and what some of the important structures are associated with the sperm. So you have three different regions. There's the head, the midpiece, and the tail. So the head contains a couple of important things, and number one, those are the chromosomes. And so what I'd like to ask you is how many autosomes are there in the head and how many sex chromosomes? So if you want to pause the video for a moment and think about that, then I'll give you the answer in just a minute. Okay, so remember sperm are haploid, and because they're haploid, that means they're 1N. That means we're going to have 22 autosomes and 1 sex chromosome. Okay, also associated with the head is the area called the acrosome. The acrosome contains the enzymes that are necessary for fertilization. And we're going to come back to the acrosome and how sperm fertilize the egg later. Then there's the midpiece, the next region of the sperm, and the midpiece contains the mitochondria. Why are the mitochondria important for sperm? So if you remember, the mitochondria are the powerhouse for the cell. So that's going to produce ATP. Sperm are motile, and they need the ATP in order to generate that energy to move. The last component of the sperm, or the last segment, is the tail. The tail is called the flagellum. And so this is what the sperm use in order to propel themselves. So when we talk about motility of the sperm, essentially you can think about the flagellum whipping, and its whip-like motion is what propels the sperm or causes the sperm to move. So how are those sperm produced? We're going to look at spermatogenesis. So this is the process of sperm production, how the sperm are actually produced. So keep in mind that sperm production is associated with the Sertoli cells, and remember that the Sertoli cells are associated with the seminiferous tubules. And so where we're going to begin is at the basement membrane, okay? And then you have a Sertoli cell here on the left. You have a Sertoli cell here on the right. And we're going to be talking about spermatogenesis that's occurring between the Sertoli cells. So we're going to start with the germ cell. So the germ cell are the spermatogonia. Germ cells are diploid, 2N. Now, remember that you have two different compartments when it comes to these Sertoli cells. You have what's called the basal compartment. So if you recall, that's from the tight junction toward the basement membrane. And the other compartment is called the luminal compartment. So that extends from the tight junction to the lumen. We're going to start at the basal compartment with the spermatogonia, and as the sperm migrates toward the lumen, they're undergoing development. So beginning with the spermatogonia, males are born with a finite number of spermatogonia. And so the thing here, though, is that these spermatogonia can undergo mitosis. And so they continually, repeatedly divide. They are dividing indefinitely after puberty. In males, they keep producing spermatogonia throughout their life. After they undergo mitosis, then the spermatogonia are going to develop into primary spermatocytes. And they do that by undergoing mitosis. So those primary spermatocytes then, they are also diploid, because whenever a cell undergoes mitosis, the cells that are derived from that are identical. So these have to be diploid cells. Primary spermatocytes then undergo meiosis 1. So when they undergo meiosis 1, they're going to produce secondary spermatocytes. Secondary spermatocytes are haploid, so they are 1N. Secondary spermatocytes then undergo meiosis 2 to become spermatids. And then spermatids undergo a maturation and development stage. So they have to mature and develop to become spermatozoa, aka sperm. So this is your gamete, the male gamete. Okay, this process, spermatogenesis, began with the spermatogonia. Mitosis took place. You produced primary spermatocytes, then meiosis 1 occurred to produce secondary spermatocytes, then meiosis 2 occurred to produce spermatids. Then the spermatids matured and developed into gametes, male gametes, spermatozoa. And all of this process was occurring as the cells were migrating from the basal compartment toward the luminal compartment. And once this process is over, the sperm are actually in the lumen of the seminiferous tubule. And I put a little note for here that approximately 50 to 500 million sperm are actually ejaculated, or that's per ejaculation.
Okay, so did you understand spermatogenesis? Yes, guys, any questions?
थोड़ा थोड़ा समझ आया मिस.
थोड़ा थोड़ा समझ आया. अच्छा, ठीक है. Whatever was there in this video, I think that's the only detail that you need to remember. In fact, if you want, you can see the same diagram on your, like in your textbook. I'll just show it to you. बिल्कुल वैसे ही description है. Just a minute. Yes. Right? This cell which you can see right at the top, जो आपको diploid नजर आ रहा है, those are again spermatogonia. Let me just highlight it for you. These ones. These are spermatogonia, and while she was describing, she was also describing some anatomy of the testes. And you know, for example, कि इस तरह से cell बना हुआ था, right? Okay, there were compartments made like this, and she was calling this central compartment as the luminal compartment and this as the basal compartment. So basically what she was explaining you was that when these cells are maturing and when they are dividing, all this is taking place in the luminal compartment. Basically, to know what the basal compartment is and the luminal compartment and the anatomy of the testis, it was a part of your syllabus up until 2014. But after that, after 2015, right, this was eliminated from your syllabus. If you go through the notes of the application support booklet जो मैंने share किए हैं, I think बीना ने मुझे कहा था on the menstrual cycle, तो आपको नजर आएगा कि उसमें anatomy दी हुई है, microscopy दी हुई है testis की. So please don't think that it's a part of your syllabus. Whatever is in your syllabus is the one which you can see over here. ये जो आपको यहाँ पे description नजर आ रही है ना, this is only required by you to remember, in which gametogenesis in the male begins from diploid cells, which are known as spermatogonia. At the top, they are showing you just one diploid cell. तो अगर आपको यहाँ पे इसका number लिखना है, आपको कभी-कभी number लिखने को भी कहते हैं, तो it should be 2n over here. If these cells are dividing by mitosis, right, they will still remain 2n. So the difference between this step and this step is that only the number of the cells has increased, which means कि पहले एक था और अब बहुत सारे, but the kind of division is mitosis, right? And then they will, after undergoing mitosis or after increasing in number, they will simply grow. Growth हो रही है, and this growth के बाद अब हम उसको spermatogonia नहीं कहेंगे, we'll be calling it a primary spermatocyte. So this means कि if I want to differentiate these steps, ठीक है, तो मैं इसको कैसे differentiate करूंगी? Just pay attention to this bit. Okay, let's say if I want to say कि what is the step taking place in this arrow, so I will say this is mitosis. Mitosis, right? Acha. During this arrow, it will be growth, as can be seen over here. Acha. What is the name of this cell? Same, spermatogonium. ठीक है, gonium कह सकते हो because it's one. And then they will divide by mitosis to produce multiple spermatogonia, as can be seen over here. These spermatogonia will grow to become a primary spermatocyte. Again, आपसे पूछा जाएगा, you will be given incomplete diagrams and you will be asked which cell has which chromosome number. So you will say कि अभी तक भी this is 2n, right? What is taking place during this time period or during this arrow? You will say meiosis 1 is taking place. ठीक है? And after meiosis 1 is complete, आप सबको पता है कि if we begin with one cell and if meiosis 1 is complete, we end up with two cells, two haploid cells. तो आपको यहाँ पे दो haploid cell नजर आ रहे हैं, right? And now you will call them secondary spermatocyte. Secondary spermatocyte, each of them, दो बने हुए ना यहाँ पे, each of them will again further undergo meiosis 2 to form spermatids. ठीक है, तो इसका मतलब ये हुआ कि वही जो हमने meiosis 1 में लर्न किया था, if we start with one cell, अगर हम एक सेल से शुरू करते हैं, if it completes meiosis 1, we end up with two cells. And if then those two cells undergo meiosis 2, we end up with four cells. This is what we've studied, ठीक है? Now the name of these four cells changes. They are no longer known as secondary spermatocytes, they are known as spermatids. And then each of these spermatids undergoes another stage. ठीक है? This is not mitosis, this is not meiosis, in fact this is maturation. Because जो sperm होता है, जो आपका adult sperm है, it looks like this, it has a head and a tail, whereas these cells are not looking like this, so they have to undergo stages of growth and development so that they look like a mature sperm having a head and a tail, right? All the characteristics, but yes, technically it's a haploid cell, which is the result of two complete meiotic divisions. हाँ जी अनस, अब समझ आ रहा है?
Yes, miss.
ठीक है, any questions? If you want to summarize आपकी understanding के लिए, so you can say कि this is a process that takes place in the gonads, और क्योंकि हम spermatogenesis की बात कर रहे हैं, so it takes place in the testes, right? It starts within the cells in the outer layer, यानी कि outer layer का मतलब हुआ कि वो जो आपको diagram दिखाया जा रहा था, that diagram, you know, इस तरह से था ना, so it was like this, right? So spermatogenesis was beginning in the outermost layer, यानी यहाँ से शुरू हो रहा था, right? And that layer is known as the germinal epithelium. If you want to summarize, you can say कि अगर इस पूरे process को summarize किया जाए, so the gametogenesis, especially spermatogenesis in specific, can be divided into three stages. One is the stage of multiplication, second is the stage of growth, and third is the stage of maturation. So in multiplication, जो है, there was one spermatogonium that was dividing by multiple mitotic divisions into many spermatogonia. And then there was a growth stage in which, you know, just growth bhi ho rahi thi, first meiotic division, second meiotic division, and then finally the maturation stage in which the spermatids were maturing into the sperms, mature sperms. I can just write down over here, spermatids maturing into sperm. और sperm का एक दूसरा नाम भी है, you can call it a mature sperm, or you can also call it spermatozoa, right? So this is basically a summary of all the different processes that are occurring in spermatogenesis. If there is any question, please do ask me. In the exams, you will get such diagrams, and you will be asked to either complete the incomplete diagrams, annotate them, or write down the name of the process. So आपको ये पता होना चाहिए कि किस stage पे कौन सा process हो रहा है, right? And also write down whether the cell is haploid or diploid. I will share these slides with you. अगर आप चाहें तो मेरी slide से भी पढ़ सकते हैं, or you can also, refer to your textbook. They are exactly the same. बस
Miss?
textbook में ये जी बेटा.
जो अभी वीडियो देखी थी उसमें माइटोसिस के बाद जो ब्रैकेट में नंबर लिखा हुआ था तो वो था 2n मल्टीप्लाइड बाय 2, उसका मतलब डीएनए रेप्लिकेशन हुई हुई है, उसका यही मतलब है ना?
जी जी जी, बिल्कुल 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 बिल्कुल। अच्छा, इफ यू वांट इट्स अप टू यू, यू कैन स्टार्ट इट फ्रॉम द, आप ऊपर से लिख सकते हैं, जर्मिनल एपिथेलियल सेल भी कह सकते हैं, स्पर्मेटोगोनियम भी कह सकते हैं, इट्स वन एंड द सेम थिंग। इन द वीडियो यू कुड सी दैट शी वाज यूजिंग द वर्ड जर्म सेल, तो वो वही बात है, जर्मिनल एपिथेलियल सेल, इट्स द सेम थिंग। और आपके टेक्स्ट बुक में ये वर्ड नहीं यूज हुआ, ओनली द वर्ड this what do you call spermatogonium has been used, so it's one and the same thing. And as far as the anatomy is concerned, so let me just show you one thing ताकि आपको थोड़ा सा अंदाजा हो जाए कि what I want to actually explain to you. अच्छा, दिस इज योर एप्लीकेशन सपोर्ट बुकलेट, ठीक है। अच्छा, अब इस एप्लीकेशन सपोर्ट बुकलेट में इफ यू रिमेंबर कि हमने जो है मेंस्ट्रुअल साइकिल यहाँ से पढ़ा था, तो जस्ट सी लुक एट द की ऑब्जेक्टिव्स। सबसे पहला जो ऑब्जेक्टिव इसमें लिखा हुआ है ना, कि टू बी एबल टू डिस्क्राइब द हिस्टोलॉजी ऑफ द मेमेलियन ओवरी एंड टेस्टिस। ये जो हिस्टोलॉजी वाला पार्ट है ना, हिस्टोलॉजी का मतलब होता है माइक्रोस्कोपी, माइक्रोस्कोपिक स्ट्रक्चर, सर्टुली सेल्स, फॉलिकल्स की डिटेल्स और किस तरह से नजर आता है, यू नो, इफ यू मेक अ स्लाइड, ऑल दैट हैज बीन एलिमिनेटेड। बाकी जो आपके सारे जो है ना लर्निंग आउटकम्स, के टू आउटलाइन द गैमेटोजेनेसिस इन ह्यूमन मेल एंड फीमेल, टू एक्सप्लेन द रोल ऑफ हार्मोन्स, बायोलॉजिकल बेस ऑफ द कॉन्ट्रासेप्टिव पिल, और जितने भी बाकी हैं, all those are included except for the first key objective. So please keep this in mind.
मिस आपका माइक म्यूट तो नहीं हो गया?
Guys, can you hear me?
जी मैं सब आवाज़ आ रही है आपकी।
आ रही है? अच्छा, ठीक है ठीक है। सॉरी, वो स्क्रीन शेयरिंग स्टॉप हो गई थी। अच्छा, आई वॉज एक्चुअली शोइंग यू, कहाँ तक आवाज आ रही थी? एप्लीकेशन सपोर्ट बुकलेट नजर आ रही थी?
मिस आपने एप्लीकेशन सपोर्ट बुकलेट लगाई थी फिर चली गई थी।
फिर चली गई थी। अच्छा, दिस इज व्हाट आई वांटेड टू शो यू कि इसका, इफ ऑल ऑफ यू कुड सी द फर्स्ट ऑब्जेक्टिव, This is not a part of your syllabus. ये जो first वाला objective है ना, to be able to describe the histology of the mammalian ovary and testis. बाकी सारा कुछ included है syllabus में. I just want to show you this. अगर आपको ये diagram नजर आ रहा हो, पुराने past papers में you will see 2014, 2013, 2012 में कि ये दिया हुआ है, तो आप सोचेंगे कि मिस ने तो हमें पढ़ाया नहीं है. This is when it has been deleted from the syllabus, so there's no need to do it. ठीक है, तो I just want to show you कि सब कुछ नहीं पढ़ना उसमें से, एप्लीकेशन सपोर्ट बुकलेट में से. ठीक है, इसी तरह से you can see the details of the detailed diagram of the ovarian follicles and corpus luteum has also been shown. That is also not a part of your syllabus. ठीक है, जो भी मैरी जोन्स में दिया हुआ है, that is a part of your syllabus, right? अच्छा, if there is no confusion in spermatogenesis, then I'll be now starting the video on oogenesis, oogenesis or oogenesis, दोनों कहते हैं इसको, so that this topic is complete for today. So please listen to it carefully.
her
It is possible that you might not be able to hear it so loudly, but you can use your earphones for that.
So let's look at the female reproductive cycle.
Okay, can you hear it?
Yes, Miss.
Acha, okay.
The female reproductive cycle refers to the maturation of eggs within the ovaries. The ovaries initially created these eggs during gestation. In other words, when a baby girl is in her mother's womb, the baby girl's entire egg supply will be created, but will remain in an inactive state. This process of egg creation is called oogenesis. Then, once she grows up a bit and reaches puberty, her reproductive cycle will start, and one egg in that egg supply in her ovaries will mature or become activated each month, and that allows it to be fertilized by sperm. By the way, another word for egg is oocyte. After an egg matures, it's pushed out of the ovary in a process called ovulation. The other major function of the ovaries is to secrete the female sex hormones, estrogen, progesterone, and one called inhibin. And we'll talk about their functions a little bit later on. So let's first discuss how the eggs are made in the ovary in the first place. So early in uteral development, precursor germ cells, which are called oogonia, and those are homologous to spermatogonia in males, these oogonia undergo a ton of mitotic divisions to make more of themselves. And then at about the 7th month of development, these divisions stop, and all the ones that have been produced, which is actually about 2 to 4 million, are all she'll have for the rest of her life. And that turns out to be about 1 to 2 million per ovary. So while she's still in fetal development, all of these oogonia that have been produced, they all develop into the next stage, which is a primary oocyte. And just remember that the two O's refers to egg, and the cyte, C-Y-T-E, refers to cell. So this just means egg cell, in case you were wondering. And let me also just mention on a chromosomal level, these oogonia, the germ cells, they're 2N, which means they have 2 copies of each chromosome, and the primary oocytes are the same, they're also 2N. And then these primary oocytes, they begin meiosis 1, and meiosis is what our germ cells use to reduce our chromosome copy number. And by that, I just mean the number of copies of DNA that we have. So they start this process of meiosis 1, but they don't actually finish it. They just kind of get about halfway through and then they stop. So they're stuck as these big cells. So they're still primary oocytes, but they're said to be in meiotic arrest. So when the female who's been developing in her mom's womb, when she's born, her primary oocytes are in meiotic arrest. So the question is, do they stay like this? And the answer is some do and some don't. Let's zoom in on this reproductive system to try to explain. So this is just a close-up of the major parts of the female reproductive system, and I've cut away parts of the uterus and the uterine tubes and the ovary so you can see sort of the inside and the outsides of both structures. And this is our key organ here. This is the ovary. So the question was, do these primary oocytes that are stuck in meiotic arrest, do they stay like that? So the answer is that the ones that are sort of destined to be ovulated, that is to be pushed out of the ovary right about here and then to be picked up by the fimbriae and then travel along the uterine tube here, those ones get past meiotic arrest. But most of them sort of die off while they're still stuck in that meiotic arrest phase as a primary oocyte. So I've mentioned the ones that sort of get out of the meiotic arrest phase and move on to develop into secondary oocytes that are able to then fuse with sperm. But when exactly does that happen? Well, it starts at puberty. So they actually stay in this phase as primary oocytes up here in meiotic arrest for like 12 to 13 years, give or take, and only then do they start moving forward with development by finishing off that first part of meiosis that they started and splitting into two secondary oocytes. And actually, that's not exactly true, even though that's what we'd expect. What actually happens is one primary oocyte, it attempts to split into two secondary oocytes, but that's not exactly what happens. What does happen is that one of the developing daughter cells develops beautifully into a normal secondary oocyte from the primary oocyte. But it turns out that when they do complete the first part of meiosis, something really interesting happens. One of these cells receives basically all the cytoplasm. So the chromosome copy number is halved, but basically all the cytoplasm is kept in one cell. So this little guy over here that didn't get much cytoplasm, it still has a full complement of chromosomes. But it still ends up being pretty small and not really very functional, so it kind of withers away and dies. And it's called a polar body. So you end up with this really large secondary oocyte, and this is what ends up getting ovulated. And so now you might be thinking, well, meiosis is 2 steps, right? When does the second step happen? And that's a good question. So again, ovulation happens roughly here with the secondary oocyte coming out. And this secondary oocyte sort of just hangs out in the uterine tubes, and a sperm comes along and fertilizes the egg. So let's look at that down here. So you have your uterine tube here, and you have your egg. That's a secondary oocyte now. And then a sperm is coming along, and the sperm fuses with the egg after fertilizing it. And so the sperm fertilizes the egg and fuses with it. And so let's just zoom in on what's happening there. So here you have your big secondary oocyte, and then you have your sperm that's sort of, let's say that the nucleus of the sperm is right here. It's inside the egg already. This is the nucleus of the sperm. And here's the nucleus of the secondary oocyte. Well, this is when meiosis 2 happens, so the second half of meiosis. So as the sperm nucleus is traveling toward the egg nucleus to create a joint nucleus, meiosis 2 occurs, and the oocyte reduces its chromosome copy number by creating another polar body, so a second polar body that kind of divides off the cell. So the oocyte cuts its chromosome copy number in half again, and so this little bit of DNA here that's just an extra copy of the DNA the egg already has, it divides off the cell in the form of another polar body that doesn't really have that much cytoplasm, just like the first one. So again, it leaves its nutrient-rich cytoplasm behind for the sperm and the egg. And by the way, the egg has changed its name now from a secondary oocyte to an ovum. But it won't be an ovum for long. Once the sperm nucleus fuses with the egg nucleus, then it becomes a zygote. So let me just clarify that if the egg doesn't get fertilized by a sperm that comes along, then it doesn't complete that second meiotic division that it did right here, and it just gets discharged from the body in menstruation as a secondary oocyte and not as an ovum, because the name ovum is reserved for the oocyte only once it's been fertilized. So those are the basic concepts behind what goes on with egg development.
Okay. Let me just make it easy for you. In females, when he was talking about spermatogenesis, the process in which when we start from spermatogonia and then primary spermatocyte, secondary spermatocyte, so ये सारा process it started from puberty. The main difference between oogenesis and spermatogenesis is that in oogenesis, this process starts before birth, even before birth, right? as you could see in the video, in which, there was a germ cell which was dividing many times repeatedly by mitosis to form millions and millions of those oogonia. And in the video, he very clearly mentioned that the oogonia were 2n. They were diploid cells. And like, what was the number? It was around 2 to 4 million oogonia are formed. And this formation starts even before birth, right? So this means that while the mother is carrying the pregnancy, for example, if the mother is about to give rise to, if a baby girl is being born, ठीक है, I hope कि आपको ये बात समझ आ रही है. We are talking about oogenesis. So this means we will have to start imagining oogenesis in a baby girl who is not even born. So this means that every girl, when she is not born, when she is in the womb of the mother, oogenesis starts at that time. ठीक है? Before birth, multiple mitotic divisions take place of the germ epithelial cell inside the ovary to give rise to around 2 to 4 million oogonia, and all those oogonia are diploid. I can say 1 to 2 million or 1.5 million oogonia in one ovary and 1.5 million oogonia roughly in the other ovary, right? Acha. And then they will keep on dividing and dividing, and now they will reach the stage where they are known as primary oocytes. So we can say that when they were in the oogonia stage, they want to enter into the meiotic division, but somehow they remain inside meiotic arrest. यानी कि वो अभी arrest में आ जाती हैं, right? And the reason why they are in meiotic arrest is that the puberty has not been reached. ठीक है, तो हम ये कह सकते हैं कि the meiosis has started, but the primary oocytes do not complete the meiosis. They remain stuck at prophase 1 of meiosis 1 throughout childhood, जो कि आपको यहाँ नजर आ रहा है, right? We can say meiosis starts in primary oocytes, which are 2n. अच्छा, अब ये start तो हो रही है लेकिन complete नहीं हो रही. Because complete नहीं हो रही, that's why I have written the word 2n. And where do they get stuck? They get stuck at prophase 1 of meiosis 1 throughout childhood until the girl reaches maturity. ठीक है? When she reaches maturity or when she reaches the stage of puberty, it is only then that the first meiotic division is completed. And when the first meiotic division is completed, यानी primary oocyte जब अपनी first meiotic division complete करेगा, then it will be now known as a secondary oocyte. In the same way as the primary spermatocyte was being converted into the secondary spermatocyte. अब आपको एक चीज बहुत गौर से यहाँ पे देखनी है कि when we were doing spermatogenesis, ठीक है, and when primary spermatocytes were converted into secondary spermatocyte, the end result was two cells which were exactly of the same size. ठीक है, sperm में जब हम कर रहे थे, लेकिन females में we will have two cells but they will not be of exactly the same size. One cell will be larger than the other cell. ठीक है, ये जो आपका large वाला cell है, you will call it a secondary spermatocyte, and the small cell is known as the first polar body. अच्छा, कोई मुझे बताएगा यहाँ पे ऐसे क्यों हो रहा है? Why is this difference seen over here in the females and why not in the males? Can someone tell me? जी.
मिस फीमेल्स में ओव्यूलेशन के वक्त सिर्फ एक एग सेल रिलीज होना चाहिए सो दैट ओनली वन चाइल्ड इज बॉर्न. अगर चार चार रिलीज हो गए तो मतलब 4 टाइम्स फर्टिलाइजेशन कैन टेक प्लेस.
Exactly. The main reason, very good, Anas, the main reason why in the female meiosis we want to get rid of this excess cell, ये जो आपका first polar body है, because अगर हमारे पास बहुत सारे सेल हो जाएंगे तो the sperms are already there in millions. It will result in multiple pregnancies every time, right? So for that reason we want to get rid of this first polar body, ठीक है, at meiosis 1, right? अच्छा, ताकि हम, अच्छा, we want to get rid of this so that we only have one secondary oocyte, और अब ये secondary oocyte जो है, it should now enter into the second meiotic division. ठीक है? अच्छा, this secondary oocyte will never complete its second meiotic division if the pregnancy has not occurred. इस बात को जरा गौर से सुनना है सबको, please. Only 1 minute is left for the class to finish. Let me complete it. ये जो अभी तक आपको ओ लेवल में पढ़ाया जा रहा था ना कि मेंस्ट्रुअल साइकिल में द ओवम इज रिलीज, द ओवम इज रिलीज, ये बिल्कुल एक गलत ही कॉन्सेप्ट था. इनफैक्ट टेक्निकली हमें ये कहना चाहिए कि इन एवरी मेंस्ट्रुअल साइकिल इफ द एग इज नॉट फर्टिलाइज्ड, तो वो एग नहीं रिलीज हो रहा होता, द सेकेंडरी oocyte इज रिलीज्ड. क्योंकि सेकेंडरी oocyte उस सेल का नाम है जिसने अपनी सेकेंड मियोटिक डिवीजन कंप्लीट नहीं की है. और कौन सा सेल नहीं कंप्लीट करता सेकेंड मियोटिक डिवीजन? द वन व्हिच हैज नॉट बीन फर्टिलाइज्ड. As you could see in the video, if the sperm is successful in reaching the fallopian tube, and if the sperm can actually fertilize the, it can enter into this secondary oocyte, jab wo andar aata hai sperm, as you can see over here, it is then only that the secondary oocyte will complete its second meiotic division. ठीक है, तो इसका मतलब ये हुआ कि to sum up we can say कि if a woman does not have any children, ठीक है, किसी खातून को बच्चे ही नहीं होते, so this means that her second meiotic division has never been completed inside her body. So it's only that when a fertilization takes place, a successful fertilization, के हम कहेंगे कि अब इस सेकेंडरी oocyte ने अपनी सेकेंड मियोटिक डिवीजन कंप्लीट की है. और सेकेंड में भी वही होगा कि जो है यू हैव एन एग और ओवम विच इज प्रोड्यूस्ड, और एक सेकेंड पोलर बॉडी बनेगी, वो भी डिजेनरेट कर जाएगी, राइट? So we end up with this and we will continue tomorrow. Tomorrow, do you have a physical class?
Yes, miss.
Yes. ठीक है. So see you tomorrow, inshallah, after a long time.
Okay, miss.
Miss, I
ठीक है.
have a question.
जी.
Miss, if in the second meiotic